All right, guys, we're going through some of the parameters that affect bacterial growth. And the last thing we did was we talked about how oxygen had an impact on bacterial growth. I just wanted to mention, along with that, something that is clinically relevant, and that would be there are certain organisms that grow in the absence of oxygen. And one of the examples would be Clostridium perfringens. This particular organism grows underneath the skin and causes gas gangrene. And the only way to treat it is by debridement, which is removing the skin to allow oxygen to get to the organism and kill it, because it's that sensitive to oxygen. It's an anaerobe. Another one is C. difficile. C. difficile lives in the gut. Most of the time, it's in the form of a spore and outcompeted by other organisms. So they stay spores and they don't cause problems, but they can germinate and become vegetative cells, and they are another example. Any of those Clostridium organisms, not only do they make spores, but they also tend to be anaerobic. So I just thought I'd mention that before launching into temperature. Okay, so we're looking now at the effects of temperature on growth. And we already saw earlier on that temperature definitely has an effect on enzymatic activity. And it would serve to reason that if it has an effect on enzymatic activity, that it would then have an effect also on bacterial growth, because they're connected, right? Without metabolism, you can't have growth. And the curves looks very similar to the curves we saw for enzymatic activity when it came to temperature. Any individual enzyme might have a curve that looks like any one of these curves you see on the slide, except here now we're looking at growth rate of the bacteria, and we're looking at different types of organisms that will grow in different ranges. All right. Now, what we're going to find is that for any one of these curves, these temperature growth curves, or effects on, curves that have an effect on how temperature has an effect on growth, I should say, you're going to see this upside down U, right? And you're going to see a minimum. So for this purple curve over here, we have a minimum that's below 0. Here with this yellow curve, we have a minimum that's around 14 degrees Celsius. Minimum for the red one around 42 or 43, right? We're going to have a minimum temperature for the growth of bacteria. We're going to have an optimal temperature, and that would be the peak. Okay, wherever the peak is, like for psychrophiles, it's around 10 degrees. Mesophiles, it's right around 30, 36, 37. Sounds like our temperature, right? So we, in fact, would probably be classified as a mesophile, okay? Thermophiles, way up there, right? We're talking closer to 70 degrees. Hyperthermophiles, man, those are way up in the 90s, right? This is what is the best temperature for these organisms to grow. And then we see a maximum. We see the top edge of what temperatures might be reasonable. For psychrophiles, it's 20 degrees Celsius, all right? So you want to be able to pick the minimum, optimal, and maximum temperatures from a curve like one of these, okay? The other thing we do is we place a name and put these organisms in categories, okay? We have phile at the end of each of these terms, so it's loving something, right? If it's a psychrophile, it loves it cold. If it's a mesophile, it likes it in the middle. Okay? Meso is, think of middle. Thermophile, well, it probably loves the heat, right? Thermo. And indeed it does. All of the temperatures here are way above what temperatures our body likes to operate at. And then we have hyperthermophiles. So we go way above that, right, with hyper, right? It's going to be much higher temperatures that these organisms like to grow at. And this would would imply that enzymes within these organisms also have a similar profile. Okay, they're going to like temperatures that are within the range that the growth is going to be best, right? Because these two are connected, right? If the enzymes are working better, they're going to be growing better. So sometimes we run into these hyperthermophiles and what we call extremophiles. Extremophiles, a lot of times, it doesn't just say thermo here. This would imply that they don't just like it hot, but a lot of times extremophiles, they like a lot of extremes, like really acidic and really hot. Or sometimes you see really high pressures too. These organisms live in the extremes of the environment. All right, let me give you a clinical example. Treponema pallidum causes syphilis. Okay, it has a very narrow temperature range. Okay, and it so happens that patients that would contract syphilis would intentionally be infected with malaria. In our past, this has happened, okay? The reason was that they knew malaria would cause a fever, okay? Malaria would cause a spike in the temperatures in the body, and this would have a tendency to kill the syphilis to some degree, okay? The other thing that this helps us to understand is when people have an organism that's infecting them, it may not be a psychrophile categorically, but it's more towards the psychrophiles when you look at its profile. We'll find that they'll grow in our appendages. Okay, so Treponema pallidum, the one that causes syphilis, the lesions from syphilis tend to appear in cooler regions. Your lips, your tongue, your external genitalia, these are places where it's cooler relative to other parts of our body. Okay. And we can see the effect of temperature on growth curves. Okay, so here we have the typical hallmarks of a growth curve. We want to be able to tell the difference between a temperature curve and a growth curve. Okay, so here we have a growth curve. I can tell because we have a lag phase. We inoculate a broth, and it takes a little while for those cells to get accustomed to the environment and start using those nutrients. We have a log phase. Now this particular log phase, in the making of yogurt, we would put this particular sample inside of an incubator. Okay, so it's going to be a little warmer in there, and we're going to start to produce the acids associated with the lactic acid fermentation, right? That's going to go on there. But then at some point, somebody has to come in and take that yogurt out of the incubator and stash it in the fridge. And you see what happens to the log phase now. That log phase is greatly diminished. It's going to grow slower because we have changed the temperature. The temperature reduced the growth rate of those organisms, and it'll stop the yogurt from getting really tart. Okay, it'd still be safe to eat, but you would kind of have this really sharp taste of the acids. We used to do this in the lab years ago. We used to make yogurt in the lab, and nobody would want to come in. We'd have a night class, and nobody would want to come in the middle of the night to take the yogurt out of the incubator. And of course, it was pretty tart. We'd still eat the yogurt, but we made sure to bring in some jam or jelly or something to mix it in there because it would sweeten it up a little bit. Okay. All right, another factor that we should think about when it comes to temperature is the plasma membrane of a cell. Okay, the plasma membrane of a cell is made out of phospholipids, and phospholipids, they have a phosphate head, right? And they have fatty acid tails. And these fatty acid tails face one another, right? Because on the inside and outside of a cell, there's going to be water. And we have this hydrophobic region on the inside. Now, if we were to look at a cell that had just saturated fatty acids, every carbon had the most number of hydrogens bound in those fatty acids, it would be saturated with hydrogens. Okay. Now, the tendency for a set of molecules like this would be like they would be all kind of bound together. It'd be all kind of situated in a clump almost. I've got a bunch of pens and pencils here, okay? So they can all be kind of packaged real tightly, all of those fatty acids, okay? And this means there will be less movement of the membrane of a cell that has a membrane with a lot of saturated fatty acids. Okay, now this is kind of like fat that we'd find on steak before we cook it. Okay, so at room temperature, there's a lot of saturated fat in the fat on a steak. Okay, and it looks solid, doesn't it? All right. So if we think about what temperature does to this kind of thing, okay? Think about butter, right? When we add heat to butter, butter changes very much so in its consistency, right? It liquefies. If we put it in the fridge, well, it's pretty solid, right? Sitting on the counter, it's kind of in the middle. All right, so if we now think about cells that are psychrophiles, okay? So a psychrophile is going to be a very low temperature, right? So at low temperatures, there would be a tendency for less movement. There's not as much heat, molecules aren't moving as much. But to have a mobile membrane, we would want to introduce more unsaturated fatty acids. So at low temperatures, we would have more unsaturated fatty acids. Okay, I know I can't write very well here with my mouse, but at low temperatures we'd have a higher number of unsaturated fatty acids in the membrane. Okay, this would allow for more movement. Unsaturated fatty acids do not look like saturated fatty acids. They have kinks in them. Okay? And these little bends in the saturated fatty acids, unsaturated, excuse me, allow for more movement within the membrane. Okay? So, low temperatures, more unsaturated fatty acids. Now, at higher temperatures, there would be a tendency for a lot of movement, okay? And we don't want so much movement that the membrane would fall apart, okay? So if we look at high temperatures, okay? High temperatures, we would see more saturated fatty acids. This will keep the membrane from falling apart, right? It won't be too fluid, right? So this is another kind of structural difference we would see based on temperatures. So where are we going to see these kinds of bacteria? Where we see psychrophiles? Well, a classic place would be out in the ocean, right? We have cooler temperatures out in the ocean. We'd see that there are organisms there. In fact, when we have had oil spills in the past, some of the news programs would cover the fact that, there may be psychrophiles. There may be oil-consuming bacteria that would help to deal with those kinds of spills. Of course, spills like those are not a natural occurrence. Likelihood is that these organisms probably do exist and probably do help to break down the oil, but we have no idea of the impact beyond that, right? Everything has everything has an effect when it comes to the environment like that. The permafrost. There are organisms that exist in the ground that remain frozen throughout the year. Permafrost is that ground that remains frozen throughout the year. It doesn't matter if it's summer or winter here in New York. There are areas of the globe where there's permafrost. Here you can see the places where the ground is continuously frozen. Now this is becoming an issue actually with global warming. As these areas thaw, those organisms that exist in the permafrost might become more active and give off even more greenhouse gases to make it hotter. Okay. Now again, something a little more on the clinical side of things. Here we have a bacterium, and this bacterium is on a microscope after having done an acid fast stain. Hopefully that triggers something in your mind, right? Acid fast, we have a primary stain that's red, carbofuscin, and it will stain stuff like Mycobacterium. That genus has species like tuberculosis, leprosy, right? Those kinds of, it's called leprae actually. So if we look at leprosy, okay, it naturally affects the appendages, okay, where it is cooler again, right? Here we can see the effects, pretty kind of gory actually, right? But that's someone's foot there and their toes have been lost because leprosy affected those particular areas. All right, we'll call it quits for now, and we'll look at some effects of pH and stuff like that in our next one.